Welcome back again to biochemistry class, and our fifth topic is about protein synthesis. Protein synthesis is basically just like the DNA or the translation process. So take a moment, take a moment to look at your hands. The bone, skin, and muscles you see are made up of cells. In each of those cells contains many millions of proteins. As a matter of fact, proteins are the key molecular building blocks for every organism in Earth. How the nucleotide sequence of an mRNA is translated into the amino acid sequence of a polypeptide protein. Protein synthesis is the creation of proteins. In biological systems, it is carried out inside the cell. In prokaryotes, it occurs in the cytoplasm, while in the eukaryotes, it initially occurs in the nucleus to create a transcript of mRNA. So, yun yung na-discuss natin na transcription of the coding region of the DNA. The transcript leaves of the nucleus and reaches the ribosomes for translation into a protein molecule with a specific sequence of amino acids. Protein synthesis is accomplished through the process called translation. Yun na nga yung sinasabi ko. So parang yung protein synthesis is like translation process lang naman. So where the mRNA is translated into amino acids, and that amino acids corresponds to a protein. After DNA is transcribed into a messenger RNA molecule during transcription, the mRNA now must be translated to produce a protein. In translation, mRNA along with transfer RNA and ribosomes work together to produce proteins. So, nagkakaisa sila. So, hindi dapat isa lang yung parang needed na raw material dito, kundi maraming raw material to produce the end product proteins. Once the messenger RNA has been modified and ready for translation, it binds to a specific site on a ribosome. The ribosomes consist of two parts, a large subunit and a small subunit. They contain a binding site for mRNA and two binding sites for transfer RNA located in the large ribosomal subunit. So, just like a book or movie that has three basic parts, we have the beginning, middle, and the end. Translation has pretty much the same three parts, but they have fancier names. So it is the initiation, elongation, and termination, which is basically just similar to the name given to the transcription process. So here in initiation, it is usually the beginning. In this stage, the ribosomes gets together with the mRNA and the first tRNA, so translation can begin here in initiation. Then we have the elongation or the middle part of the process. So in this stage, amino acids are brought to the ribosome by RNAs and linked together to form a chain, specifically a polypeptide chain. And lastly, the termination part or the end part. In this last stage, the finished polypeptide is released to go and to do its job in the cell. So, to discuss it further, During translation, a small ribosomal subunit attached to a mRNA molecule. At the same time, an initiator tRNA molecule recognizes and binds to a specific codon sequence on the same mRNA molecule. A large ribosomal subunit then joins to the newly formed complex. The initiator or tRNA resides in one binding site of the ribosomal called the P site, leaving the second binding site or the A site open. When the new tRNA molecule recognizes the next codon sequence of the mRNA, syempre nag-a-attach siya to open the A site. A peptide bond forms connecting the amino acids of the tRNA in the P site to the amino acids of the RNA in the binding site. Next is the elongation process. As the ribosome moves along the mRNA molecule, the tRNA in the P site is released and the tRNA in the A site is translocated to the P site. So, yung yung tRNA in the P site daw is nare-release na after the ribosomes moves along, and yung nasa A site naman is na-translocate or nag-moves dun sa P site. The binding site becomes vacant because of the parang translocation or until another tRNA that recognizes the new mRNA codon takes the open position. So, since there is a polypeptide chain, so hanggat di nag-enter dun sa di tayo nagre-reach dun sa stop codon, tuloy lang yung process and it will be translated into protein. This pattern continues as molecules of tRNA are released from the complex, new tRNA molecules attach, and the amino acids chains grows. And in the termination process, the ribosome will translate the mRNA molecule until it reaches a termination codon on the mRNA. Usually, ito yung tatlong stop codon. When this happens, the growing protein called polypeptide chain is released from the tRNA molecule and the ribosome splits back into large and small subunits. Now, the newly formed polypeptide chain undergoes several modifications before becoming a fully functioning protein. So, nagkakaroon muna siya ng several modifications bago maging ganap na isang protein. As we have a variety of functions. Yung proteins ay maraming variety of functions. So, some will be used in the cell membrane, while others will remain in the cytoplasms or be transported out of the cell.